Hi again. So now we will talk about topic 2, organization of the organism. In this topic, we will talk about the cell structure and cell adaptation, and we will learn about the term cell, the level of organization. So we will start with the cell, group of cells forming what, group of tissues forming what, and so on. And finally, we will compare between the structure of plant cell, animal cell, and bacterial cell. This will be for the first part. The concept map will help you to make sure that you know everything in the structure and you know everything related to the typical structure of a particular cell. Even if we are talking about animal cell, you will be able to compare between animal cell and plant cell. If you will talk about plant cell, you will definitely differentiate between plant cell, even in your mind, between plant cell and bacterial cell. So let's go directly through the presentation. As you know well, cells are the basic building units or building blocks of life. If we imagine the cell, we should imagine the cell as a factory. It has a lot of machines, and each machine or worker definitely a part of specific organization and is doing a specific job and specific function. We studied the cell by studying two types of microscopes. Microscopes can see the internal compartment of the cell, the inner structure of the cell. If we understand the cell, we understand life itself. If you are able to understand cell and cell structure, you will be able to understand life itself. So light microscope is a type of device can magnify objects, and this magnification up to 1500 times. However, electron microscope also is an electric device, and it can magnify objects to more than 200,000 x. So what is the difference between both? It's not a matter of the difference in our course, but nice to understand that we can study any metabolic activity or any kind of movement for a living organism by using the light microscope, but electronic microscope to study the deeply internal structure of the cell, because we cannot study any living structure using electron microscope for a reason. Electron microscope needs a tube pumped out of air, so we cannot study any living activity or any activity belong to a specific living organism using the electron microscope. Now, we can see the animal cell and plant cell under microscope. Simply the simple ways of differentiation, or the simple point of comparison, that the animal cell has a regular shape and the plant cell has a regular shape. Plant cell has a lot of green pigments called chlorophyll inside a chloroplast, but animal cell has no chlorophyll or chloroplast. Let's go through the structure. This is the structure of the animal cell. And animal cell has a nucleus with the genetic material in, has chromosomes. Chromosomes are the genetic material that carries all of our characteristics. The nucleus has a nuclear membrane, and this nuclear membrane has some pores, and the pores are opening just to communicate, to send some signals from the nucleus to the organelles in a structure, gel-like substance called cytoplasm. So cytoplasm is the structure that carry out all of the chemical reaction of the cell. Mitochondria. Mitochondria is an organelle responsible for releasing energy. And please don't say produces energy. Okay, so releases energy is way better, as energy is neither created nor destroyed. Cell membrane to control what enter and leave the cell. Ribosomes for protein synthesis. And vacuoles. Vacuoles are either few or absent in animal cells. If we have some vacuoles like this, it will be really tiny, but it's few or absent in animal cell. For our course, I recommend to say absent. There is no vacuole in the animal cell. Look at the structure and all of the description and function for cell membrane, cytoplasm, and the nucleus. Let's go to the plant cell. For the plant cell, we have cell wall, and I can see now the plant cell is regular, has a regular shape or more regular shape. It has a lot of green structure or green organelles containing chlorophyll, and we name them chloroplast to carry out the photosynthesis process. I can't say that we have any other differences except the vacuole. Vacuole is big, large structure found in the middle of the plant cell, and it contains all of the water, or it stores all of the water and minerals in the cell. Okay, so this is the typical shape of the plant cell and animal cell. You cannot imagine, guys, how CIE examiners love this drawing. So very important to know that we have a lot of chloroplasts, and please differentiate between chloroplasts and mitochondria in your drawing. The chloroplast is like that. It's a lot of thylakoid or stacks of granum like this. And the mitochondria has a zigzag-shaped structure or inner membrane like this one. Okay, let's go for the all of the structure, and actually that's what we explained in the previous slide. So to save time, we can talk about bacterial cell. What makes up a bacterial cell? Bacterial cell, unicellular. It means that that it made up of only one cell. It has a cytoplasm, ribosomes, cell wall. Look, to cut it short, nothing on Earth living has no ribosomes. Ribosomes responsible for making proteins, and honestly, we cannot live without protein. Plasmid or section of DNA, and we will talk about plasmid in the last couple of topics, because this is a genetic material in bacteria, is super beneficial for humans, and we will talk about this later. The DNA, as we said before, is circular DNA. It's a circular DNA, not linear DNA like the animal cell. Okay, that's all what we mentioned about bacteria. It has cell wall. Yes, it has cell wall made up of murine or this type of carbohydrate. So if you want to differentiate between animal, plant cell, and bacterial cell, this table has been designed for you to summing up everything related to the differences between all of the cells. The second part of the lesson related to levels of organization. As I told you, cell is the basic unit of structure and function for all living organisms. What about group of cells? Group of cells forming a tissue. So in the exam, if he asked you to define a tissue, you should say group of similar cells performing a particular function or working together performing a certain job. A group of tissues form an organ. And now organ made up of group of different tissues working together performing a certain job. Organ systems, it's okay, group of organs working together to form the whole body. And finally, the whole body is made up of a lot of organ systems working together. So let's take a tour on the next couple of slides, and then I will show you the part of levels of organization. Now, the question is, we are close to finish topic 2. The question is, how is the structure of a cell related to its function? Look, we have some specialized cells. Not all of the animal cell guys or plant cell have the same structure. No, some cells has been specialized in order to do a specific job. Let's take a look on some plant cell like palisade mesophyll cell and root hair cell. The palisade mesophyll cell is a cell found in the leaf or the layers of leaves for photosynthesis process. So it is elongated with a lot of chloroplast. Look, a lot of chloroplast and the chlorophyll in all of the chloroplast for photosynthesis process. So this is an adaptation. Root hair cell. The root hair cell has been adapted by having this long and narrow root hair for absorption of water and mineral ions. So each root cell has, or the majority of root cells have some root hair, and this root hair for more absorption of water and mineral ions. For in animals or the human body, we have different types of cells has been adapted to do a certain job. The first type of cell is the red blood cell. Red blood cells has been adapted to transport oxygen. Okay, so it has cytoplasm containing chemical substance made up of protein called hemoglobin. And it has biconcave shape in order to pass easily through tiny blood vessels or capillaries. It has also no nucleus to provide more surface area for taking more oxygen to be transported to the respiring tissue. Eggs and sperm cell. Eggs are the gametes or sex cells for female, and those gametes has jelly coat, nucleus, and cytoplasm. And we will talk about the structure of the egg cell in topic 16. For the sperm, it has head, middle piece, and tail. The middle piece has mitochondria because sperm has a hard trip in order to reach the ova, to fertilize the ova in the female's body. So it needs a lot of ATP or large amount of energy to be released from mitochondria. It has also a specific area in the head called acrosome, and the acrosome responsible for dissolving the membrane or this layer of the egg. So it has acrosome and this acrosome dissolving by specific enzymes the membrane of the ova. Ciliated cells for movement of mucus in the trachea and bronchi. Nerve cell, it is a tree-like structure responsible for transmission of electrical signals. So it has an axon, and this axon insulated with myelin sheath, because, as you know, our body is a good conductor of electricity, and during transmission of signals, we are talking about a lot of electrical signals and electrical ions. That's what we mentioned, guys, about the tissue, organ, and by the way, we can find a lot of examples from plant cell, animal cell, but the criteria is totally clear. If you have group of similar cells performing a certain job or specific function, this means that you have a tissue. If you have group of different tissues or one or more kind of tissues working together to perform a certain job, this means that you have an organ. And don't forget that the leaf of the plant is an organ. The organ system is made up of several organs working together for a common purpose. An organism made up of a lot of organ systems working together. Last part is extremely important for both paper 2, paper 4, and paper 6, or paper 1, paper 3, and paper 6 for core IG. Very important to know how to calculate the magnification of any specimen. And our formula is I am formula. I am formula. Magnification equal image size divided by actual size. Instead of memorizing this as a traditional boring mathematical equation, you can keep the triangular structure in front of you all the time. So under any circumstances, you can answer any question related to magnification, and this will give you 3 marks in paper 6 and might be 1 mark in paper 2, and from 1 to 2 marks in paper 4 if you found a question related to magnification. But most probably you will find the questions in paper 2 and paper 6. Okay, look at this question. Figure 2.17 shows the transverse section of a tomato fruit. The actual diameter of the tomato fruit is 50 millimeter. He wants you to calculate the magnification of the tomato fruit. So he's giving you A, and he wants you to calculate the magnification. So all what we want to measure from the image is the diameter. So you will draw a line here, and you will draw a line here, connect both using a ruler, and measure the diameter. Now, if you have the diameter in millimeter, and you have the actual length in millimeter as well, so you can definitely find the magnification. Look at the answer. This is the image size divided by actual size, so you can get the magnification, as you can see. The last part of this question, please, for all my year 9, year 10, and year 11 students, very important to know how to convert from millimeter to micrometer. 1 millimeter equal 1,000 micrometer, because in some questions he enforced you to get the answer or to write down the answer in either micrometer or millimeter. So if he wants the answer in micrometer, multiply by 1,000. If you have all of the known or given in micrometer and you want the answer in millimeter, divided by 1,000. Now, we have covered topic 2 as well. So more videos are coming for all of the topics of the syllabus.